Okay, can somebody hear me? Can anyone hear me?
I can hear
Yes.
you.
Great,
Can hear
thank
you.
you. Um, I don't know how to show me or anything on the table, so I'm just gonna have to do my best today. Um, it is true, I do need to mention that I may be serving jury duty next week, but I don't think it'll interfere with the time of this lab, meaning I think 6:30 is late enough. I don't think it'll be a problem. Let me share my screen here. My computer is going to be a bit slow because it's still converting the meeting. Where are we at? We're at 87% from the lecture. So bear with me a little bit. Let me share my screen here. All right, you should be seeing my screen now. Before we begin, are there any questions? All right. Today we're going to cover Lab 01, the ubiquity of microorganisms. Remember, the worksheet is due at 8 a.m. this Friday, January 10th. If you do not have both the worksheet for Lab 01 and 00 in, I will drop you from the class. You also need to read the infectious disease research project, which you can find on Canvas. Let me show you. It's under the week 2 lab. So read this infectious disease research project by next Tuesday. And don't send me a species of choice until Tuesday. I will not take it until Tuesday because the project isn't starting until Tuesday. Let's see. And then remember, you have to take the online plagiarism quiz by this Saturday, 11:59 p.m. on Saturday, the 11th. It is true if you don't get it in by Saturday, I will extend it to another week, but you should really try. I've had two students who had trouble getting the quiz to run, and I know one of them, probably both of them, they checked out computers from the Clark College, probably a laptop, and they'd already had Respondus Lockdown Browser on it, as well as Respondus Monitor. And when the students downloaded the new programs, the new programs wouldn't work, meaning to get the lockdown browser and the monitor to work, you have to use the version that Clark College has already put on the laptop. Any question about any of that? Oh, good. The lecture Zoom lesson just finished, so my computer will now start working much faster and better. All right. Any questions about any of that?
I just have one question.
Yeah.
So the practice one that's due, I think, this week or next week. What is that one? It's only 1 point and it's like 13 questions, I believe. I just want to be prepared for anything.
That's a practice exam. It's not due this week. I think it's due next Saturday. And it is to get you prepared to take my exams. I'm going to give you up to 1 point of extra credit. You'll get about 0.1 point of extra credit for each correct answer. I thought it only had 10 answers, but it might be 13.
I couldn't remember. I just was wondering. I just wanted to make sure. So is it just based off of some of the questions you'll be asking for the first test?
No, practice quiz 00 covers the syllabus, labs 0 and 1, and then chapter 1. Quiz 1 covers labs 0, 1, and 2, chapter 2, the start of chapter 2, and then chapter 1 and 10. So they do cover different material, although similar, that sort of overlaps, but um, it's a little different. Anyways, on my syllabus, I've got what the quizzes cover, what you need to know for the quiz. So I've got even on the practice quiz. I don't have it on the plagiarism quiz, but it's sort of a different quiz. And to tell you the truth, I haven't taken the plagiarism quiz for many, many years, so I don't remember what it even covers. But it covers the material in the plagiarism tutorial, how to correctly cite information, when you should cite information, and then how not to plagiarize. That's what the tutorial covers.
Out of curiosity, if you've taken that plagiarism, the test quiz a couple of times, um, once you take the Respondus version, will that little green check mark finally go next to it if you get through that finally?
I'm not sure what you're talking about, so I don't know how to answer the question. But let me state you do have to take it for this class, even if you've taken it again, meaning you do have to take it for this class. So if you've had it several times, sorry about that, but it should be easy if you've had it before.
Mhm.
All right, let's go to the lab module 1, ubiquity of microorganisms. There is some reading to do in each of the labs. This just a few pages from the textbook on culture media, pages 157 to 162. You do need to read about a page on obtaining pure cultures. There are some video clips in this lab. The learning objectives for this lab are: one, know that microorganisms are almost everywhere on the planet Earth. Describe the purpose of culture media. Two, three, describe what colony abundance and colony morphology mean. And then four, be able to define the following terms. What's inoculation? What do we mean by the ubiquity of microorganisms? What's something that's sterile? What is isolation? What is incubation? What is in vitro, like in vitro growth? What is a culture media? And we have different types of culture media. We have a broth that is a liquid culture media. And then we have a agar solid media, and that we call usually an agar, but it could be solid from something other than agar, like starch or gelatin. And then we have different tubes. We have a slant and we have a deep. I guess I'll show you those later. What's something that's fastidious? You probably should have something. What is something that's not fastidious or non-fastidious? What is a non-selective media? What does ubiquitous mean? And what is a colony? Any questions about any of that? All right. Microorganisms are organisms that are too small to be seen with the naked eye. It does include viruses and prions in this group, even though really they aren't organisms, they don't have cells, and they're not, well, at least some people don't consider them alive. We're not going to get into that argument there. We also include helminths, worms, parasitic worms, in our studies of microorganisms, even though the adults are not microscopic. However, their infective stages of the microscopic worms usually are microscopic. What does the term ubiquity of microorganism mean? It refers to the concept that microorganisms are almost everywhere. It's not entirely true, but they are almost everywhere. Something that is sterile is something that's free of living cells. Like when we give you a sterile tube of media, if you were to come into the real microbiology lab, it would be sterile. How we get it sterile is we put it in the autoclave. In this lab, we're going to investigate the abundance and diversity of microorganisms in common environments, and you're going to learn some basic lab protocols or procedures as well. A culture medium, also known as a growth medium, is a sterile laboratory preparation that contains a variety of nutrients that are required for growing microorganisms in vitro, meaning in the lab. We can have a culture media that's a liquid. These are called broths. They're commonly put in a test tube because if they're put in a plate, it'd be just too easy to spill it and then contaminate the culture. You can add agar or some other solidifying agent to the liquid medium to result in a solid medium. Agar is often grown in a petri dish, but it may be grown in a tube as well. We have a little video for you to watch in preparing pour plates, so click the link there. Note how the culture medium needs to be sterilized prior to its use. An important question to consider is, why must a culture medium be sterile prior to it being used? So think about that. I guess somebody could answer if they wished. All right, here we have 3 tubes of media. That is a liquid culture that we call a broth. These 2 are solid cultures. This solid media is called a slant because it's formed in a slant. This one is called the deep, where you simply pour the liquid in, allow it to cool, and then it solidifies. And that we call an agar deep. The petri dishes are usually a solid media in it, like an agar media, meaning it's too easy to spill if it's a liquid broth in a petri dish. Today, we're only going to talk about one type of media. There are different types of media. Today, we're going to talk about a general purpose media. It's non-selective in function, meaning we do not select for certain cells to grow in the media if it's non-selective. General purpose media are usually complex in their composition, and they support the growth of most non-fastidious organisms. A fastidious organism is one where you must add specific ingredients to the media to get the organism to grow. So non-fastidious, they're easier to grow. We generally grow non-fastidious organisms in a general purpose media, and there are 3 that we need to talk about. The first I'm going to talk about is number 3, nutrient agar, or Na. It's a very simple general purpose media containing really only a couple of things, mainly Roundup beef extract, sodium chloride, and water. It does have something else in it, but those are the main ingredients. Tryptic soy agar is something similar, except it's using protein that you get from soybeans, why it's called tryptic soy agar. And the abbreviation for nutrient agar is Na, the abbreviation for tryptic soy agar is TSA. And then we have nutrient agar with yeast extract, or Na. It's a little richer than nutrient agar because it's nutrient agar that they have ground up yeast put in, and the yeast supply some nutrients. Anyways, these are 3 very common general purpose media that we will be talking about all term long. Any questions about any of that? All right. In a microbiology lab, labeling is very important. All petri dishes and tubes must be correctly labeled prior to use. The following 5 items should be included on the label. You should have your name, the date, the type of medium like Na, the inoculum, what you're going to be growing in the culture medium, meaning what you're going to be growing. Like here on this one here, we're growing E. coli. If it's something like you don't know what it is, like bacteria that fell out of the air, just say something like air exposure. And then the incubation temperature is optional. You may include it. It doesn't have to be on the plate. But the name of the person should be on the plate, what you're growing, the date, and then the media. All of those are required. They should be put around the um, periphery of the bottom or the base of the plate, or you can put it on the side of the plate. You don't want it in the middle of the plate because it'll interfere with your ability to see the growth on the plate. Any question about any of that? Tubes, you normally write this information on a piece of tape and then stick the tape to the tube. About that, I guess I'm going to talk about it here. When we grow, I guess you can't see that, so it's hard for me to show you because I can't show you, but I'll go through the actions even though you can't see it. Sorry about that. But what I'm got here is a petri dish. I'm taking off the lid. You can't see that, so I'm trying to describe it. In my hand, I have a pretend loop. And when we put something on the plate, which I just did, cover the lid back on, we do not stick the plate in the incubator right side up, meaning where we have the top lid on top of the plate. What we do is we have our growth and then we take the plate, turn it upside down, and then put the plate in the incubator upside down. The reason being is that if you put the plate in right side up, moisture would come out of the agar and it would simply evaporate, and then it would condense on the bottom of the lid until a drop forms, and then the drop would fall down onto the agar, which contains your growth, and that would disperse your growth. So instead of having isolated colonies, you'd have a whole bunch of growth growing in between the colonies, and so you wouldn't have anything isolated. Another reason is that if the water tends to come down the side of the petri dish on the lid, when you're taking the lid off of the petri dish, the drip can come down the side and then fall into the culture, which would contaminate the culture with unwanted organisms. Now, that's only going to happen under those conditions. Normally, when the drip of water drips down from the lid, it would be sterile because the bottom of the lid would be sterile. So contamination would likely not happen, but you increase the risk of contamination happening. So it's not really for contamination, it's just to keep what you're growing separate and not growing all together and unisolated. And if you put it in the incubator upside down, the moisture is already at the top of the plate. And so if it comes out, where it's going to form is on the drop would form on the agar. And even if it were to drip down, which it won't, we'll talk about that in a minute. If it were to drop down, it's going to drop on the bottom of the lid, and you have no bacteria growing on the lid, so who cares? In actuality, the drop would be wicked by the agar, so a big drop would never form. You probably wouldn't even get a small drop because it'll be wicked by the agar, and then the moisture would be dispersed throughout the agar plate, so it would not drip. Any question about any of that? So the important thing is when you put plates in the incubator in microbiology, you put them in upside down so that your cultures do not become a messed up heap of cells growing all together. By inoculation, we mean placing or transferring the microbial specimen into a sterile culture medium, either a tube or a plate. And then you take that tube or your plate and you put it in an incubator. Incubation is the placement of an inoculated culture medium in an appropriate physical environment for a prescribed time to allow the growth of microbes. Normally, in microbiology, you grow microbes in an incubator, but you don't have to. You could grow them at room temperature. And that would not use an incubator. And I won't talk about that, but um, incubators can be complex, as shown by this picture here of an incubator. Many different dials here that can control the temperature, can control the amount of oxygen in the incubator. Meaning you have to shut the door to get it. This has two doors. I'm not seeing the second one, but maybe that's one there. A glass door there, and then the outer door. And it can also control the amount of CO2, the amount of humidity, and then the temperature, and of course the time for how long you allow it to grow in the incubator. Usually for humidity, what you simply do is you get a dish like this, put water in, and then uh, the water would evaporate, bringing humidity into the air, making the incubator humid. That's actually important because with a petri dish, it would only stay about 3 or 4 days in an incubator without added water, meaning humidity, before the agar would dry out and then the cells would begin drying and dying. If you put water in the incubator, making it humid, the plates will stay at least a week. Sometimes you can get them to grow for 2 weeks. Any question about that? Most of the incubators actually all of the incubators we have in our microbiology lab at Clark are simple incubators, and they only control the temperature, the humidity, and then the time you grow the cells in the incubator. They do not supply oxygen and carbon dioxide. You're simply using the oxygen and carbon dioxide that are present in the air. All right. When we have colonies growing on an agar plate, we look at the plate, we can see little bumps on the surface of the agar. Each of those bumps is a colony. Colonies can have different characteristics. They can be rather smooth or rough looking. Colony can be at least 1 million cells. A colony under 1 million cells is too small for the human eye to see, so all colonies must be at least 1 million cell. Most colonies are between 10 and 100 million cells in a colony. All of the cells in a colony could be a descendant of the original cells that started the colony. So if one cell started the colony, all the cells in the colony would be derived from that one single cell, meaning one cell would divide into two, two into four, four into eight, etc., until you get 1 million. Here's a little video, real short, showing you how cells can go through binary fission to become one cell to eventually grow into a colony. It's a real short movie. In theory, one cell leads to the formation of one colony, but you should realize a colony may be formed by two or more cells. It could even be formed by a clump of cells. Sometimes if you see difference in colony size, like this colony is bigger than these two colonies and much bigger than those colonies, these are probably all the same species. Not that one, but all of these and these three, probably the same species. This colony is probably bigger. I don't know for sure, but probably bigger because it was formed by more than one cell, probably by a clump of cells. These colonies are bigger than those colonies because they're isolated and simply because they have more resources. But that one is actually less isolated, so it should have fewer resources, but it's actually bigger than these two. So my guess is that was formed by a clump of cells, and these by one or very few cells, and these probably by one cells. Any questions about any of that?
I do have a quick question. Will you be
Uh-huh.
testing us on some of this stuff that you're talking about in
Yeah,
this
of course.
lab? Okay, perfect. I just want to make sure I wasn't writing down things that I didn't need.
Okay. Yeah, this is lab one, and you will have quiz questions from lab one. So remember, when we look at a colony, it may be formed by one cell, but about 30% of them are formed by two or more cells. And it really depends on the species for how many are formed by one cell. Colonies are not visible in a broth culture because if you're growing things in a broth, where do I have that? All the cells will mix all together. You don't get growth in a colony. I've got this someplace. Oh, there we go. All the cells will just grow and mix all together. And if you have more than one species in there, you won't be able to distinguish them by their colony characteristics because there will be no colonies. Colonies, when grown on a solid culture, which is the only way you can get a colony, is they will have visible colony morphological variations or differences between different species of bacteria. Here we're looking at a table of colony characteristics. We have a shape. That's the overall shape of the colony. You're going to have a circular colony, an irregular colony, a spindle colony, and then rhizoid or filamentous. I'll never ask you to distinguish between is this colony rhizoid or filamentous, but it could ask you is it circular or irregular, or rhizoid or filamentous, meaning is it circular or is it one of these two? There's the margin, that's the edge of the colony. A circular colony has an entire margin. Usually the colony would be irregular in shape, have an undulate, a lobate, or a curled margin. I'll never ask you, is the margin undulate or lobate, but you should be able to distinguish these three from an entire margin. And then I'll never ask you, is this margin rhizoid or filamentous? But it could ask you, is it one of these two, one of these three, or this one entire? Any question about any of that? All right. For elevation, you have to really take off the lid to the plate and then look at the colony from the side to see the elevation. We can have a flat colony. We can have a raised colony. That's sort of like a mesa. You can have a convex colony. You can have a pulvinate colony. That's just an extreme convex. I'll never ask you, is this colony convex or pulvinate? Because I would have a hard time distinguishing these two. And then there's an umbunate colony. Let me show you what we're going to deal with here. Are these colonies flat raised, convex, or umbunate? Anyone have a guess? When you're looking at a photograph, it's impossible to tell the elevation of the colony. Like I said, you need to look at the colonies from the side and then determine, is it flat? Is it convex? Is it raised? Let me see if any of these are raised. Yeah, whoever did this wasn't looking at the original plate. They're looking at the photograph, and so they don't say the elevation because you can't. Well, that one, colony B, that one
Is
they say is
colony
flat.
A convex?
It's hard to say.
Okay.
Like I said, you need to take a look at the plate with the lid off and at the side to determine that. The colony has a different size. I can't show you the pencil, so I'll have to just tell you. If the size is punctiform, that means the shape of the colony is smaller than the point of a dull pencil. Not a real sharp pencil, but smaller than a dull pencil. If the colony is small, it's about the size of a dull pencil. If the colony is moderate, that means it's between the size of a dull pencil and the size of the eraser on the pencil. And if the colony is large, it's larger than the eraser on the pencil. Uh, you can see the texture in the colony. The colony can be smooth or the colony can be rough. Students often get a hard time distinguishing that, and I'm sure it would be difficult, especially on a picture. But colony B, they're saying it looks rough. When I looked at it, I'm not entirely positive of that. Let me see if I can blow it up. Probably rough, but you can't be certain on that one. You can be certain that is not shiny. That one is shiny, so that you can tell. That's our next colony characteristic. Blow it up a little. Whether the appearance is shiny or glistening, or whether the appearance is dull. And colony, was it B? Colony B is a dull colony. Colony right here, not labeled, is a shiny colony. Colony A is also shiny. Colony C is a good one that's shiny. Doesn't say it, but it's shiny. But I can't tell for sure that's convex. It's saying it's convex. And I can't really see about the smoothness, but we'll go with that. All right. Let's go on to the characteristics. The last two is pigmentation. If the colony has any color to it, we say it is pigmented. Let me blow this up quite a bit. All right, that's kind of brownish. We don't have any good colored ones here, but that's kind of brownish, orange brown there. Let's move up to here. These are yellowish, so is that one. So these are pigmented because they have color. The white, that one, off-white, close to white, grayish, light colored. All of those are non-pigmented, and they don't have to be white. They don't have to be off-white. They can be any of those, I should say, lack of color.
Question for you.
Uh-huh.
The fungus up in that upper photo you were just showing us, it's kind of black or possibly brown in nature.
Uh-huh.
Would you consider that pigmentation or not really?
If it's got brown, which I don't see, then it's definitely pigmented because brown is a color. I'm not sure if black is considered a color or not.
Okay.
Well, I'm not sure if it's pigmented or non-pigmented. Most colonies are not black, and
Yep.
that's the only one I've ever seen that was black. Of course, it's a mole. It's
Right.
black.
Okay.
So I'm not sure. And when it was smaller, that probably didn't have black in color. But I'm not sure if black is considered pigmented or not. So I'm just not going to answer it. I'll tell you the outer rim was white or whitish or off-white. That's non-pigmented. But I don't know about black, so I'm not going to mark you down. I'll never even ask, is black pigmented or not? Because I'm not sure. Black is really the absence of color, and white is all colors mixed together. So, you know, I'm not sure. It's definitely not red, blue, or green or yellow. And let's just stay with that for pigmented. And black, I don't know. I'll have to do a search on that sometime because I don't really know. It's funny, this is the first term I've ever thought about that. Don't ask me why, because we had that black colony for a long time, but oh well. And then the last property is the optical property of the colony. Most colonies are opaque. A few colonies are translucent. Very, very seldom have I ever seen a transparent colony. A transparent colony looks like a drop of water on the petri dish, meaning you can see right through it. They're actually hard to see, but translucent, we do have a few here, not many. The best one is this one here, where this portion of the colony is translucent. And I don't know if that's another colony on top of this one, or that's the center of this colony. The center looks opaque. That's opaque. That's opaque. The edges here look translucent, only a little bit, slightly, but the center looks opaque. Any question? That's clearly opaque. Oh, there's one. There's a very good one. That one is that translucent right here. Two terms you need to know is what is colony abundance and what is colony diversity. Colony abundance refers simply to the total number of colonies on an agar plate. Way too many for me to count there. Not even going to try. Here, I'm going to estimate that this would be between 30 and 50 colonies. It'd be really hard to count them right in there, too, and right there. But if we look at just these region of the plate, the colony abundance is only 3. The colony diversity are the number of different colonies on the plate. So on this plate here, in this region there, what is the colony diversity? Anyone want to guess?
3.
Yeah, that's got different characteristics than that one, and that's different also from that one. So right there, the colony diversity is 3. On this plate, that's probably the same as these. So that's still 3, 4, probably 5, 6. A little white one, so that's 7.
Are you looking for exact numbers like on our lab?
No, as long as you
Okay.
have something close.
Um, when the colonies end up touching each other, like say in the very top center there where you've got the three or so colonies kind of jumbled together,
Uh-huh.
that's considered a single colony, correct?
No,
Like, no? Okay.
no. You would count that as 3 colonies because it's very clear those are 3 colonies, which at one point were isolated when they were small, but now they've grown together. Now, this one here, is a clump of colonies. I'm not sure how many there would be in there. The same with that there. So when you're counting them, just do your best. I won't give you something that's that hard to count because this would be impossible to count the colonies there, there, and even here. That's clearly two different colonies, but there could be more than that. And that one, I'm not sure what's in there. But no, this one right there, count that as three.
I wanted to ask, when you were asking about how many, are you considering the full plate itself?
And
I mean, I know,
normally, like when I ask for colony abundance, we're referring to the total number on the plate. And I didn't want to take the time to count these,
mhm,
but I'm guessing it's between 30 and 50 would be the
I
colony
see.
abundance for this plate. I'm not going to give an exact number because it's going to take me too long to count. And it'd
Okay.
be impossible to count these there and there's those there too.
So, just like on our lab, when you're asking for the
Ju-
abundance, it's
Just
just a
give
rough
a
estimate.
close number. You don't have to be exact.
Okay, thank you.
Okay. As long as you're close. Actually, let's go back to that other plate. Colony diversity is the different number of colonies on the plate. And this will be a little bit open to interpretation, but I would say these are the same. So all of these are the same. So that's one, there's two, that's probably three, four, five, that's the same there, six, seven, eight, possibly nine, that little one. That's probably the same as that one. That's probably the same as that one. So I'm going to say this one about nine. 8 or 9 would be a really good answer. If you give something under 5, I'll tell you it's not close enough and mark it at least partially wrong. And if you were to give something like, I don't know, diversity over 20, that would obviously be wrong also. So your numbers should be close, but they do not need to be exact. And another reason for that is there's difference of interpretation. What is the same species? Like that right there, is that a small species colony of this one? Are those the same? I can't tell you. These right there look a little different to me just because of the coloration and the size, but they're similar. And it's possible this is just a darker, smaller version of that one. So in diversity, there is a little bit of opinion about it as well. But if you're close in your numbers, you'll get correct points. All right, here's a real world example. What is the abundance of wild cats in this picture? How many cats do you count?
7?
Yeah, seven's correct. And what's the diversity of cat species?
2.
Two. Cheetahs here and lions there. That's a male lion and a female lion, if you're wondering why they look different. In case you don't know, most of you should know. And let's see, there's 5 cheetahs right here and 2 lions. For the abundance is 7. In this case, the diversity of cat species is very easy, but with bacteria, it's not quite as easy. All right, you know the terms of this lab? Some cute pictures of a hand. You don't need to see the references. I think I cut off too much here. Up. We're going to be doing everything on the computer, so obviously you can't do this directly, but you should know the general concepts and the general ideas of this lab. And procedure 1, we're going to be asking the question, what is in the air you breathe? Below is a nutrient agar plate that was exposed to the air and then incubated for 24 to 48 hours. And procedure 1, question A is, what is the colony abundance on this plate? Just count the numbers, total numbers, put the number there. As long as you're close, you'll get full credit. If you're way off, you will get it wrong. What is the colony diversity on this plate?
I would say
A little
5.
harder question. Just a minute. That could be the same colony species. That's definitely different. That could be the same as that. So that's at least 2. I'll count minimum. That's reddish. So that's 3, 4. That could be the same as that. 5. Did I count that one? I don't remember. And that little one, 6. This funny one, 7. Those could be the same as that. I've already got a little one, so I won't do that. 7. Could be more. Like if you count these as separate from that and that. And then on step procedure 1C, while you are inoculating a plate, meaning you take off the lid, get your loop, inoculate the plate, put on the loop, not the loop, put on the cover to the plate. What can you do to reduce airborne contamination of a sterile agar plate while you are inoculating it? Do not tell me what you can do before you begin inoculation. Do not tell me something you can do after you've inoculated it. So tell me something you can do to reduce airborne contamination while you are inoculating the plate. Any question about any of that? All right, procedure 2. What gets on your lab benches when you put your lab bench on the table? Here we have a inoculation of a lab bench before the student disinfected the workbench, or they put disinfectant on and then sterilized, well, not sterile, cleaned it, and then removed the worst of the disinfectant and then allowed it to air dry. So that's beforehand, and then that's afterwards. And question procedure 2, did table disinfection work? That's a yes or no. You can state a reason if you want, or an explanation. And then 2B, what is the purpose of allowing the table disinfectant to air dry? Or why do we air dry the disinfectant? We could take clean paper towels, for example, and totally remove the moisture from the disinfectant so the table would be dry, at least to the touch. But we don't. We just remove the moisture of the disinfectant and allow it to air dry. Why do we do that? I don't think that's explained in the lesson, but you could get a question and answer online. And if you use AI to do that, you should give a quote where you got it. Because otherwise it'd be plagiarizing the AI. Procedure 3, what is on your hands? Here we have a person who touched the plate with his fingers. There's the fingers there. Probably a thumb there. And then that's the left hand here. That's the palm. And there's the fingers going down. And then they washed their hands, dried their hands, and then touched the plate again. You need to dry your hands so that your comparison will be similar to the hands before they're washed, which presumably were dry. The reason is because if you have moisture on your hands, you will wick the bacteria from your hands to the plate. And the first year I was teaching microbiology, I noticed that students, when they actually did this in the lab, were getting more growth on the, or at least some students, more growth on the plates where they washed their hands. And I asked them eventually, did you dry your hands? And they always said, oh no, I didn't want to get contamination on my hands from the paper towels. And I told them, okay, that explains why you have more bacteria from your clean hands than your dirty hands. And that's because your clean hands were wet and the moisture wicks the cells from your hands onto the plate. Even if your hands have much less bacteria than before you wash them, the moisture will pull more and wick it to the plate. Any question about any of that?
Can that be a similar answer for the above question
No,
for
because you're
B? Oh, okay.
inoculating, you're swiping a counter
I see. Okay.
before and after. All right, the questions for procedure 3, does hand washing work? You can say yes or no. You can also give an explanation there. Meaning maybe you don't want to say it didn't totally work, but I think it worked partway, something like that. Doesn't matter, just yes or no or give an answer. And then 2, suggest one reason why hand washing did not result in the complete removal of all microbes from your hands. Meaning we do see some microbes, and it may have to do with the normal microbiota on your skin. It may have to do with the soap that we use to wash our hands. For example, if this is using normal hand soap, what would happen if we wash our hands with regular hand soap and then put our hands in chlorine bleach? We'd probably see fewer bacteria cells, if any bacteria cells, on the clean hand compared to the hands before we washed them, meaning the dirty hands. Anyways, on this question, if you get some information, you must provide the source, meaning online information. Especially provide the source if you get it from AI. Procedure 4, when you kiss your significant other, what exactly are you giving him or her? And here we have a petri dish where somebody is kissed. There's the lower lip, there's the upper lip, and then this is where somebody touched their tongue on the plate. And for these pictures, you should rate the note that I added above the picture. And then for procedure 4A, which plate has higher colony abundance? The lips or the tongue? And then B, why do you think this is the case, even though you might expect the abundance in both body sites to be the same? The lips is right next to the tongue. You probably frequently lick your lips with your tongue. So why is there a difference in the abundance between the lips and the tongue? And for this answer, you must give 2 reasons to be correct. It's very easy coming up with 3 reasons for why one plate will have more than the other, but it will be, uh, it's possible to come up with 5 different reasons. I mean, I've actually had students come up with 5 different reasons, but 3 of them are very easy. And to get this answer correct, you must use 2 reasons, at least 2. Any question about that?
Is that just for A, or do you mean
No, just for B.
for
You
B?
have to have
Okay.
2 reasons why there's more colonies on one of the plates than the other. Okay? All right. C. Why was the tongue plate incubated at a higher temperature than the lip plate? I didn't mention that, but we come up here. It says the lip plate was incubated at 30 degrees C in an incubator, and the tongue plate was incubated at 37 degrees C in an incubator. Why the difference in temperature? Procedure 5, for microorganisms in the soil, what we do is we set up a tube of water, usually 10 mils, and we add something like 1 gram of soil to the tube, and then we take a dilution of the soiled water and spread it on the plate. And when we did that and got it to grow for 24 to 48 hours, we saw this plate. And procedure 5A, what is the colony abundance on this plate? And 5A2, is it even possible to accurately answer this question? Please explain your answer. Any question about any of that? Procedure 5B, is there a greater abundance of colonies from the soil compared to the lip plate? So there's the soil plate, there's the lip plate. Some students have said in the past that there's more colonies on the lip plate because there's lots of little colonies on the lip plate. Let me point out to you that there are, and they stated that there are large colonies on the soil plate. Let me state that there are more large colonies on the soil plate, like right there, right there, right there, right there, right there. These are large colonies. There are some like large colonies on the lip plate too. 1, 2, 3, 4, 5, 6. There are more large colonies on the soil plate, but there are a tremendous number of small colonies on the soil plate as well. So right in there, we have a whole lot of little colonies. Right here, so many little colonies grown together, you can't count them. Right there, so many little colonies grown together, you can't count them. I guess I'm sort of giving you an answer there. Sorry, didn't mean to do that. But so do realize that the soil plate has a large number of little colonies as well as a large number of big colonies. So going by the colony size is not a good way to answer question B. Is the greater abundance of colonies from the soil compared to the lip plates? And then answer question B. Any question about this lab? All right, it's about 7:30. I'll be here until 8 o'clock to answer any questions you may have. Remember, this lab will be only graded one time, so you should think about your answers and give a correct answer, because I will read everything. And if you don't have the correct answer, it will be marked down for being at least partly wrong. So I guess if you like a lab instructor who doesn't read what you turn in and then they just accept anything you write, I'm not the instructor for you. Okay, I will read everything you write.
I do have a question about the worksheet for lecture one.
Uh-huh.
It looks like it looks a really just chaotic. We have one that's like 1 through 8, and then it jumps to 19, 18, 17, 16.
All right, you're talking about the worksheet in the lecture?
Yes, worksheet lecture one.
Okay. Yeah, I'm
Um,
aware that students have not done the directions, not followed the directions, and they
right,
jumped all over the place. It makes it really difficult when you do that to determine what is the next question to answer. So please
exactly.
always follow the order of the worksheet. And if you don't like that question, just don't answer it and come back to the worksheet at a later time. Someone will answer that question for you, and then you can take the next available question. Because if you don't take the next available question, it makes a mess of the worksheet, and it's almost impossible to figure out which questions have been answered and which questions have been answered and what questions have not been answered. So the first
Exactly.
person who did it wrong will be penalized. But because so many people did it wrong, I understand, I'm not going to penalize anyone other than the first person. So
Okay,
I'm
so
being a
my
little
question,
generous to everyone else because it got all thrown out of whack. Just do your best, try and find a question that's not been answered.
it
If
looks
all
like
the
all of
questions
them
are
got
answered,
answered. Yeah.
if all the questions have been answered, try and find a question that you feel is answered incorrectly or at least partly incorrectly and elaborate. And if
Okay.
you find that all the questions are answered correctly, just start all over again answering the questions in the order they come in the worksheet. So
Okay.
you would answer question number one, and the next person, if they didn't find a question they thought was wrong, would then answer question number two.
Okay, that was my thought process as well. I know the last number was 4, so I was thinking about doing 5, but 5 has already been done multiple times. And I've been kind of going over looking at each one, seeing if there is one that's incorrect, but so it's
Okay.
just a little chaotic. So I'll re-look at it and then
I think somebody has already restarted the numbering, but I don't know. I haven't looked at it.
yeah, the last one I just because I was re-looking at it right now, the last one is number 4, but it went from 3, 13, 4. So looks
Okay.
Right, and
like
the reason
Well, I'm
reason
glad that
why
somebody
there's
did 4, that somebody tried to reestablish the order. And if the
yeah,
next person would have done 5 and 6 and 7, it would have reestablished the order and almost everything would be correct. But if you take the questions out of the order, in the future, everyone will get a penalty who takes it out of order. But
right.
since everyone did it incorrectly, or not everyone, but a lot of people did it incorrectly this time, it sort of made a mess of it. I'm only going to penalize the first person who did not follow the directions.
And whoever it was number 19, and then people did 18, 17,
It
16, and then we
doesn't
moved up to
matter. I'll take a
13.
look. You don't need to tell
Yeah,
me. Yeah.
but I was just trying to figure out, do I need to start from 5? Because
Yeah, what
number
you should
4
do
did
is
it.
find one that has not been answered or one that you feel is answered incorrectly. And if all of them have been answered, just start all over again.
Sounds great. I will do that.
All right.
Thank you.
All right. If there's no further questions, I will be here to help you until 8 o'clock, but I want to stop the recording and I will be here to answer questions. If ever I leave, I will tell you that I'm leaving and I'll be back in about 2 minutes. And I'm going to have to do that because you can't see me even when I stop the share, which I think I've stopped the share. Yep. And we're going to have to do it that way until I get this settled as to what's not working. I didn't change anything this term. It just started where it's not showing me. And I don't know how to change it. It must have been an update in Zoom. All right, get started on the lab in case the question is of interest to someone else. Okay, go ahead and ask your question.
Yeah, it's super simple and it might be a dumb question. I just really want to make sure that I'm learning the material and doing everything correct. Um, I know this sounds like really simple to me, but does this work for an answer? It's procedure to um letter B for what is the purpose of allowing the table disinfectant to air dry? I said the purpose is to expose the microbes to the disinfectant for amount of time it takes to for them to kill the microbes. Would that be an okay answer?
That's an okay answer.
Okay, perfect. I just want to make sure that I'm going down the
Work on my laptop.
Did you check out the computer or laptop from the school?
No, this is my personal.
Well, then you need to contact the computer hub or hit the help button in Lockdown Browser and get
Okay.
them to troubleshoot it.
Okay. I
Probably
will.
you need to change a setting on your computer, probably
Okay.
just one, so that it'll work.
Okay. I know my camera's just broken on it.
The
So
camera is broken.
yeah, I'm getting a new laptop.
If the camera is broken, you're going to be in trouble.
Okay.
You need the camera for the uh, exams. But the thing is,
Yes.
you can email me personally, and I will, after everyone's taken the exam, I can, if you don't have a camera, because you say it's broken, you know it's broken for sure.
It hasn't been working, and I'm gonna take it in tomorrow into Apple so they can kind of figure it out.
Okay, well, hopefully they can fix it.
Yes.
Okay. Do you have a recent version of your Apple?
Yes, I do.
Okay. Yeah, hopefully they can fix it then.
Perfect. Thank you so much.
All right.